Our next speaker is Dr. Girinath G. Pillai, sir. I welcome you, sir, to grace the event on the topic. Sir's topic is computational approaches in drug delivery, drug discovery, and precision oncology. So, starting with his brief introduction, Dr. Pillai is one of the coordinators for Government of India MHRD Drug Discovery Hackathon 2020. Congratulations, sir. Director and founder of ZASTRA Innovations, Bangalore, visiting scientist at Nairo Research India, and formerly a Marie Curie Research Fellow at Molport Limited, Estonia. He carried out his Ph.D. at University of Florida with Professor Katzinski and Professor Carlson, University of Tartu. He holds 40 research papers in peer-reviewed international journals, one US patent, and co-authored a book chapter on mosquito repellents. His expertise is in the area of computational medicinal chemistry, mechanistic driven drug discovery, and modeling software, pipeline development. KNIME workflows attracted global projects from the ExxonMobil, USDA, Johnson & Johnson, and Eastman US. Currently, he focuses in the area of aging and longevity studies. Apart from his scientific positions, he is also a mentor of IIT Koetlam, Government of India, Niti Aayog ATL School, and research advisor to VIT, UPES, and other institutes across the country. I welcome you, sir, to join us. Please, sir.
Thank you so much, madam, for the kind introduction. I hope I am audible.
Right, sir. Clearly audible, sir.
Let me try to share my screen. Just a second. Okay. Yeah, okay. Now I can hear you. Okay. Can you see my screen in full screen?
Yes, sir, it's visible, yeah.
It's visible, but not in full screen, sir. Yes, sir, it came.
Okay, great.
It came. Thank you, sir.
Yeah. So before I start, I want to just have an understanding. Just all the faculties joined here are all of which background? Just to stress on specific things if there are very specific areas are focused. So just to understand that.
Sir, most of them are from pharmaceutical sciences. Some biotechnologists are also with us.
Okay.
And pharmacy students, PhD candidates.
So it's a mix of everyone. Okay, great. So some slides will be for chemists, some slides for pharmacologists, some slides for biologists. So so if there is something that you are not able to follow, please let me know. I know it's only 1 hour we have. I'll try to cover at least the initial phases. And I want it to be more interactive. That's where I confirm that this is really useful to the participants or the attendees here. So we are here to learn each other, right? Anyway, so thank you so much, Rupa madam, for this kind opportunity, including all the organizers as well as the management of NIT. And thank you all for this. Yeah, absolutely. My pleasure. So moving forward, we will today look at the computational approaches in drug discovery and precision oncology. Why, when I say computational approaches, everybody has in mind, yeah, bioinformatics, sequence analysis, and then finally docking. That's it, done. But there is way beyond that. So today, I'm not going to talk about all these bioinformatics, docking, or all those things, because that is somewhere it's already very much prevailing and known to everywhere. But I want to say something, how these drugs came into the market just using docking or something else also has to be done. So we need to know the reality. And I'm very happy that all the attendees we have from different disciplinaries. Drug discovery is a multidisciplinary area. Just a chemist cannot come up with a drug. A biotechnologist cannot come up with a drug. Or even a bioinformatician cannot come up with any hypothesis. We need all of them together on a single table. And then only this works well. And that's why I'm always jealous and I feel bad why I did not study anything related to or M Farm or B Farm, why I did not do my studies earlier. I focus more into chemistry. Well, good, but still, I could have got into something more interesting at that time. So today I will be more focusing my agenda. It's not like the topic of my slides. I'm trying to say what areas I'm going to focus, how to help or how to make decisions on which molecule I have to work on. How can we make a call that, okay, I have to work on compound B, not on compound A. So that methodology perspective will be given today. Then why do we require digital transformation? I understand there are a lot of biologists, experimentalists here in the attendees. So we need to understand why do we really require computational predictions? And I'm agreeing that, and most of you agree to me that experimentalists also use computational studies. The computational studies that you use is mostly statistical analysis. Without statistical analysis, I'm sure there is no experimental results that you're going to predict because you need to understand the experimental data variability, standard deviation. So ultimately, statistics has its biggest role there. But when we say a bit more of a computational study, we say structure-based analysis or fragment-based or ligand-based analysis. And let me tell you, fragment-based, ligand-based, or structure-based is not bioinformatics. Bioinformatics is completely sequence-based, proteomics, and genomics. And once it reaches to structure, once the structure is being elucidated, the role of bioinformatics is done, right? After that, the chemist comes into the picture, pharmacologist comes into the picture. If you ask me, bioinformatician cannot do this, yeah, you can do, no problem. But chemist understands much better about the chemical structures, and I will say why in a moment. Then we also need to know workflow automation, because as I said, it's a multidisciplinary. Multiple people are combining to do with a single objective. So we need to know what is a transfer of knowledge from one expertise to the other expertise. And finally, lead optimization cases. So I have 3 lead optimization cases, especially on anti-cancer drugs, which has come into the market with the help of structure-based drug design, and which I will be discussing that today. So just a single marketing slide. I am coming from Shastra Innovations. We deal with packages for drug discovery, drug development, pharmaceutics, statistical thermodynamics, GraphPad, I'm sure everybody knows that, and many more others, including Cosmotherm and API co-crystal screening. And also we look at with the solutions to innovate with parallelization. So moving forward, we want to start with how to guide or how to make decisions based upon certain information that we get computationally. Now, why do we need that computation or digital transformation? Because we need different viewpoints. As again, I'm repeating, we need to have knowledge transfer, being it's multidisciplinary. So different viewpoints has to be looked at. I might be having 20 years of experience, but I'm always going with a different viewpoint. But a person who recently came might be having a different viewpoint. The techniques learned will be having a different viewpoints. Of course, science is still the same. Perspectives lead us to explore the unexplored things. So limited experiences, I myself proudly tells you I'm not an experienced person in this area because I'm daily learning it. Without daily learning, I cannot say that I'm an expert. So I'm having a very limited expert. So even within our own field, we have limited experience. We cannot be mastered in each and everything. If I say chemistry, am I mastered in chemistry completely? No, maybe in a very specific niche area. Limited memory. I might have read 10, 15 papers, but I don't remember everything. I remember only a fraction of it. But computers can remember and recall much more than what humans can do. I don't deny humans are much more intelligent than computers, but memory recovery or recall or experiences, that's also we have to look at. And that's where we wanted to understand what is the difference between information and knowledge. We have a lot of data available in the textbooks, YouTube, wherever you see, training slides. You might be listening to the slides and then you might get inspired if you might me, but that is a data for you. But when these things are being applied on your projects, when you try to implement this on your project, that's where it transforms to a knowledge. So information is like a paint in a tin. You never admire that color. But when any color that is being applied on a canvas, then you admire it. Oh, wow, this color is really great. Awesome. So knowledge is like a paint. So computers can apply the stored knowledge much faster than a person because computers can give you different perspectives. Now, you might have done docking a person whom they have taught you. But if you follow the same pattern, how they taught you, you're not exploring new things. Docking can be done in 100 different ways. The results can be interpreted in 100 different ways. If you did not try that, you're not looking at different perspectives. So that's what computers can help you. Of course, it will not tell you whether it is an agonist or antagonist, but at least it can give you some data to help you to make that decision. Reason is, scientists have hidden biases. We choose which knowledge to believe, and we are very conservative and constructive also, right? So computers can explore ideas more quickly and rigorously than a person. But with the biggest disclaimer, if you do it in the right way, don't think that giving everything on the computer, it will give you the right results. No, garbage in, garbage out. So learning denotes changes in a system that enables a system to do the same task more efficiently the next time. And this is where I wanted to mention a particular quote from earlier head GSK. The grand challenge in our life is to how best to incorporate existing knowledge. We say that this is being published 100 times. I'm also going to use the same method, same technique. But did you realize to understand what was the mistake or what was the challenge in that methodology? So that's where we have to learn. And when we are trying to improve it or enrich it, that's where we wanted to optimize it. So when I say in a chemistry terms, okay, you already have a compound with you and you understand its activity, you understood its toxicity. Now you understood which region is having problematic. So as a chemist, you wanted to make modifications because you learned something that is from the enabling the system to improve its efficiency. And that's where you make modifications in the functional group to improve its activity. Similarly, that's what we call it as machine learning also. Or from statistics, we understand a trend and next time we may improve it. So this is applicable starting from an infant baby till to any rocket science. We learn from our mistakes, we learn from our knowledge, we learn from existing data, and we try to enrich or improve next time when we apply it. But if you're replicating the same thing, we're not doing anything new. That's why many of the journals are now not accepting many papers because they don't see novelty because it's exactly the same protocol being published in 100 papers. It's just the difference in the disease, just the difference in the compounds, right? So this is something that we have to see how efficiently next time you can use it because we are going through an era called Bio 4.0, right? Even it is in the pharma or even if it is in the biotechnology sector. And I don't know how many of you are aware that we say that GDP in India is great in research, pharma and biotech sector, about 32%. But have you anyone realized how much is the intellectual property staying back in India or for India? It's just 8% of the GDP, even less than that. The rest, huge majority of the contribution of the GDP comes from service industry, that is contract research. We work for others and they take the IP, they keep the IP, we get the money, we get the job. But if a country cannot sustain IP, that means intellectual property in pharma and biotech, that's not good. And that's where the basic research has to become much more stronger. So these data, whatever I told you based upon 2019 might be improved. I really don't know. I'm waiting for the reports. So why do we bother with calculations or simulations? Because we want to accelerate research, reduce cost, environmental impact, and reduce experimental search space and analyze structure property reactivity. But disclaimer, if you do the computation in the right manner and in the right direction. And that's where we need to understand the accuracy level, because we are comparing chemistry, biology with empirism. So accuracy will differ. And do you think that experimental wet lab experiments are more accurate? No, there are still errors there involved. And that's where we have to, when any experimental data comes, we ask them how much times you have repeated it and what is the standard deviation. Synergetic experiments, because we have to ask relevant questions. Whatever that questions you can ask in a wet lab experiment, don't try to ask the same question in computational. They are different. So there should be a synergetic effect or synergism between computational and wet lab studies. And are we going for finding only the best ones using computational studies? No. If you say a best target, a best structure for anti-cancer, we cannot find them in computational studies. Rather, we can find a group of compounds or a bunch of compounds which could be a candidate, but with further validations. So for a software, in order to call how these softwares can be made or what are the requirements of the software, it's not that highly published software is the best software in this world, nothing like that. It's not that if you use this software, you can publish papers. I think you have to take away from that concept. No reviewers are asking, why didn't you use Autodoc to publish? Or why didn't you use paid software to publish? No, it's your vision, how it is computationally efficient, how it is validated and robust. That's what we have to look at. So this is what I was trying to tell you, the workflow automations, where we can try to plug and play multiple things together when multidisciplinary comes into the picture. But I want to tell you something here in the drug development process. I'm sure everybody is bored of seeing these kinds of slides everywhere. But here I want to give a different message. When you're trying to do your wet lab studies, maybe you might be bringing in 2 compounds or 5 compounds for your cell-based assay or even for animal studies. Do you think 5 or 10 compounds is enough? Now, the biggest blame will be resource or funding. When you're asking for funding, ask for 100 compounds. Let them give you compounds, at least for 20 compounds or 50 compounds for testing. Without that, we cannot translate our basic academic research to a commercializable research. That's very important. So the development time and the money, even when they're starting in the phase 1, we still have 20 compounds there in the drug development process. And it goes on the funnel reduces and you can see 5 to 10, 2 to 5 compounds. And then it finally gets into 1 to 2 compounds with a cost of more than 1.1 billion pounds. And finally, when the compound gets approved, it's almost 11 to 12 years, many thousands of subjects being tested, research volunteers, and finally we come up with a compound. But still, the research will be ongoing. Now, you might tell me, sir, based upon AI and ML, I think that we can get the drugs in the market 3 to 4 years. Yeah, we pretend to be, or we are expecting it to be. Let it come, then I will update my slide. But it's too much hype here and there for AI and ML in drug discovery. Many things are happening, but whatever the publications are coming up, many of them, I don't say all, many of them are not reproducible and not reliable when you look at the physical meaning of it. Of course, the model looks great with R squares and other statistical parameters. But there's a biggest problem that we have to solve, even in anti-cancer drugs also. I hope everybody knows about imatinib, right? And I don't know how many of you are aware, the company who produces imatinib is Novartis, and they give these drugs for free for the desired people, so that at least they don't miss their courses even if they do not have money, right? So the drug regulatory authority has to approve that this person is eligible to get that medicine for free. But the reason is it's not affordable. So we need to know why there's an increase in the cost of medicine and how we can reduce the cost of the medicine. That is very, very important. Many of them, they don't take medicine just because it's not affordable to them. It's mainly because of the cause of failure of oral drugs. Between 1960s and 1980s, based on these two reviews, it said that most of the oral drug failures was because of poor pharmacokinetic.
Dr. Soumya, is there any problem? Sir is not audible.
Yes, ma'am. I hope so. From the sir side, sir is not audible.
Hello, sir. You are not audible at this moment.
Sir is in meeting, but voice is not coming. Okay, now leaving.
Mr. Obijit, will you please call sir once? He is not audible.
Yes, ma'am. I'm trying.
Yes, PPT is also not visible now.
I have just spoken to Dr. Girinath Pillai, sir. He's trying to reconnect. We have to wait for a few minutes.
Okay,
He's
okay,
trying
thank
to
you.
reconnect. Yeah, sorry, speak.
So can you hear me and see my screen?
Yes, sir.
Okay. Sorry about that. My network went down.
Sir,
I
the
just
slides are
reconnected it.
not visible somehow, but you are, yeah, yes, sir, yes, sir, it's visible now.
Oh, okay, great. Okay, fine. Sorry about that. My network was down. I think now it shouldn't be an issue. So I'll repeat it. So the cause of major of the oral drug failures was because of the poor pharmacokinetic ADME properties and efficacy also. I have an example today for anti-cancer drug also where it was having poor pharmacokinetic properties and they had to optimize and improvise it. And of course, that is already there in the market once it is being optimized. Now, that's where the role of chemist comes into the picture. So later, between 2000 and 2010, the failure of drugs reasons were something different. It's not pharmacokinetics or ADME because we were able to reduce that impact by implementing computational tools as well as quantitative structural activity relationship. And that was really gave a boost to reduce the failure of oral drugs. But later we have a new problem, that is the strategy and toxicity. We were being using different sophisticated methods and we were messing it up and the compounds are becoming more toxic when compared to earlier drug failures. And that's where when we look at the new medicine phases, it always starts from basic research. Exactly what I do, what you do, and many of you do. And then it translates to drug discovery and preclinical. I think at least few of you will be getting at least to the stage of preclinical. Very few, I don't say many. It has a lot of funding involved, a lot of people involved. So at least to get into preclinical. Once it is IND submitted, and that's where you go for a patent. And this is a translation that every academia has to look at. How can we convert our basic research to a preclinical and at least to get it patent so that a commercial entity will attract to it and they require the data, what is that they wanted in your patent for the technology transfer. Now, let's look into the discovery phases where and when, which people and which expertise is required. When we go with the drug discovery, we always have to start with target identification. If a structure or a target is already being explored, and that's where the place where we need biochemists, biotechnologists, and bioinformaticians to understand on which mechanism, how the genes are associated with certain diseases or disease associated variations we wanted to understand. And we also wanted to understand to identify the right key amino acids which is responsible for upregulation or downregulation of certain mechanisms of enzymes. So these target identification process is not that easy and it's very important. So if this is very clear, where you can always compare with computational studies with your wet lab or bioassays, here comes the lead discovery and optimization. We need to identify which could be the possible drug molecule or drug candidate or a small molecule which can actually inhibit the malfunction on a very layman terms that I can say for a target to correct it or to inhibit or to activate it. But then later we found the activation is all fine, the binding is all fine, but later I found it's toxic. But how should I overcome that toxicity? I cannot go with another compound because I found this compound is really active. So that's where the chemist comes into the picture to say that, okay, I can optimize, make modifications. I can make modifications and come up with a better compound here. And then we look at the ADME toxicity, PBP case studies, and more simulations comes into the picture. And then, once all good, we get into clinical trial after certain regulatory filing and applications. Luckily, all these, whatever I've shown here, can be assisted by computer-aided design. This does not mean that computer-aided design is a replacement to wet lab experiments. And never try to do that. And never try to claim in that way. The computer-aided design is helping you to make decisions, helping you to reduce the size in the funnel. That's all. But ultimately, you have to do the wet lab studies to make validations. If you ask me the success stories of computer-aided design in the pharma, there are more than 112 molecules and many molecules already in the market. And these are something is in the phase 3, phase 4 trials, which is still yet to come, and some are already in the market. And if you want to have a little bit more about the list and all, you can refer to these two references which I have cited in this slide. But we have a data overload challenge. Too much analysis, too much results, and we try to get confused with what we have to get at the end of the day. Initially, we select some few compounds to do MTTSA if we have many compounds. And whichever we found it is active or inhibiting, and then we select those compounds. If it is not, we remove them from our data set. And later we try to do some other tests like absorption rate test or metabolic stability of these compounds. And then each step we are removing certain compounds because it is not viable or it is not stable or it is not active. But ultimately what we expect is that we are getting a very good compound because that's what we presume. But unfortunately, 95% of the cases, this is not what is happening. This is a filtering approach, what we always try and we usually do. So when you try to see that, even though your compound is highly potent, it may be chances that they might be having a very poor absorption or maybe very toxic. In that case, the most active compound you have thrown out. And this is called as a missed opportunity. Sometimes we use filters, like Lipinski rule of filters, unnecessarily in wrong places or wrong stages. And this is what it happens. And ultimately, you end up with a moderately active or non-active compounds. So this strategy itself has to be looked at and relooked so that you to be very cautious when you're trying to apply these kinds of methodologies. Because the data comes from in silico, in vitro, in vivo for the same set of target, for the same objective diseases and few set of compounds. So there we have to prioritize based upon the importance which property has to be taken more, given more important. The biggest importance has to be given to the activity itself or potency itself. And that is the most topmost priority. And later, when you try to select the compound, make sure you give a balance between quality as well as diversity. Because when we look at only at quality, there will be less room for optimizations. So I'll just skip this slide due to the interest of time. So I'll quickly get into the main theme of this FTP program. So whatever so far I have given is just a groundwork to give you a basic overview about what all things we have to take care when we get into drug discovery. Now, when you go to anti-cancer, I'm already aware that you already gone through 2 days of sessions, and tomorrow we have Raghava sir and few more other speakers who is going to discuss more about a little bit more of different aspects. So I'm giving an overall overview with some examples. So we are focusing on abnormal cell as well as a malignant cancer. But this all the reason when you look at it can be from heredity, UV radiations, chemicals, cell dividing, smoking, as well as viruses. But then the risk of cancer in men and women is also slightly different in certain organs and certain cases. I'm not going through that because you guys are already aware of that. If you look at the cancer types in humans, there are plenty of types, and they are all target specific also. So these all pictures I've taken from internet. I don't own any copyrights. It's all their own property. I'm just using it for the education or awareness purpose. So when you look at the classification of anti-cancer drugs, I'm sure all of you are aware of this because of the different cell cycles stages. We have hormonal drugs, anti-metabolites, topoisomerase inhibitors, and microtubule inhibitors, which gets into a list of all these drugs which we have. Drugs acting on cells, which is mainly of cytotoxic drugs, which are alkylating agent. Also, we have platinum coordination complexes. We have anti-metabolites, which are like folate antagonist, purine antagonist, and pyrimidine antagonist. Then we have camptothecin, taxanes, Vinca alkaloids, and monoclonal antibodies, including hormonal milieu, antibiotics, and tyrosine kinase inhibitors. I'll be discussing about one of the kinase inhibitor today, about VEGFR2 inhibitor. So there are several inhibitors and different classifications of anti-cancer drug where you can start if you're working on certain new set of molecules or a new set of derivatives. So the topoisomerase inhibitors are camptothecin analogs, and microtubule inhibitors are the taxanes as well as Vinca alkaloids. And there are many other anti-tumor antibiotics. Platinum coordination complex I have listed in the top. Then tyrosine kinase I have listed, including monoclonal antibodies. There are very few selected ones. Yeah, you can search papers and online, you will get a wider list of data there. Now, so I have picked up three main drugs, which has been derived from computational studies using especially structure-based drug design, and they did not find the structure directly from a docking study. They had to optimize it. So this is where I'm going to discuss today. So first I'm starting with Cristoninib, which is a gene amplification against lung cancer, lymphoma, and esophageal cancers. This is very selective and potent against c-Met and ALK dual inhibitor, and which was approved by FDA in 2011. This is completely based upon structure-based drug design, where lipophilic efficiency was employed as a parameter for the binding effectiveness. So lipophilic efficiency they had looked at and enriched it. CMET, which is also known as HGFR, which is hepatocyte growth factor receptor, and its corresponding natural ligand, hepatocyte growth factor, which play a critical role. So we were looking and focusing on the overexpression of CMET protein, which has been often detected in human cancers. Abnormal function of CMET signaling was observed in various solid and blood tumor cancers. On the right hand side, you can see the protein. Oh, I forgot to mention the PDB-ID. I'll just look at it. So we are looking at the structure of the human anaplastic lymphoma kinase in complex with cristinib. So you can just search this particular wording, structure of human anaplastic lymphoma kinase, you will get the PDB-ID. So the structure you can see here, this structure was derived from another structure, so optimization was taken care, right? I'll show you in a slide where the optimization scheme was taken care with this particular compound. Now, the next one is axitinib, where it's a therapy for advanced renal cell carcinoma. On the right-hand side, you can see the chemical structure of axitinib, and the target here, the protein structure is the structure of VEGFR2 kinase domain in complex, and the PDB-ID is 4AG8. So this is also again based upon structure-based strategy and served as an inhibitor by binding to VEGF kinase domain in the DFG outconformation. And this also signaling plays a crucial role in the development of malignant diseases. So two important hydrogen bonds with the hydrogen bonding groups from these two amino acids at the position 885 and 1046 of VEGFR2 provided highly potent inhibitors. And this is the key part which I was telling you about target identification. Because before forming your structure-based drug designing or docking studies, you need to have an understanding which are the key amino acids I have to target. Which are the key amino acids I have to expect some interactions. If we do not have an idea about that, we cannot say which compound can be called as antagonist and agonist. But most of the time, what we have read in papers, based upon the docking, they will say that this we found using docking, the compound is having interaction with these amino acids. No, we need to know beforehand the biochemical or mechanism, which are the key amino acids. So axitinib, its a marketed name is, or commercial name is Inlita, in combination with pembrolizumab, or I don't know, I think Keytruda, was approved as a first-line anti-cancer drug against renal cell carcinoma, which is RCC. This also is a derived and optimized drug. Another one is lumnifsib, which is a strong heat shock protein anti-inhibitor, and which is now in the clinical trials. And HSP90 has become a promising target for cancer treatment. Of course, it's already in the market. And also heat shock protein has 4 functional domains, which is a middle domain, an N-terminal domain, ATP-ADP binding domain, and a C-terminal dimerization domain. And it's completely based upon high throughput screening. They were able to identify this compound. On the right-hand side, you can see the heat shock protein. Its PDB-ID is 6LTI. And the compound structure is given in the top. But the biggest thing that I wanted to discuss is how did we reach to these 3 compounds? That's where the role of medicinal chemist or pharmaceutical chemist comes into the picture, right? Understanding the target, understanding the mechanism, receptor, all biological people, pharmacology, all played a vital role there. But now we need to understand where we can enrich. All these 3 compounds started from a different compound. And then they got into finally a marketed drug. How did we achieve this? Number 1. Number 2, how did we know that this particular region has to be modified? So this slide is mainly for chemists actually, but still it will give you some insights for the others why your compounds are failing in wet lab studies. So we need to understand which region of the molecule has to be also modified. And this investigation started with evaluating the series of three substituted intolerant 2-1s, a potent class of kinase inhibitors, that is intolerant 2-1 derivatives for c-Met inhibition. Among the derivatives, the first one, PHA 66572, showed strong activity against c-Met autophosphorylation process and the corresponding biological activations both in vitro and in vivo. So activation, all your potency studies were all good with the first structure, which is there in the A. But however, bad drug-like characteristics on that compound limited its further study. The co-crystal analysis of this compound with kinase domain of c-Met elicited the key inhibitor binding site, presenting the opportunities of efficient drug design. But later, with the substitution with the newly designed derivatives, with this displayed a promising inhibition against c-Met, and it also noted that lipophilic efficiency was employed as a parameter for binding effectiveness. Later, we were able to carry out applications of structure-based drug design based upon the initial studies to improve its drug likeness parameters or characterization, where what it has been done is the optimization of benzyl oxy group, the functional group at fifth position. So basically this one, the fifth position of the two aminopyrimidine and examination of the chiral center, the cristonibib formed, which has an effective tumor growth inhibition and a good drug performance as being able to get achieved. So this actually demonstrated a remarkable clinical efficacy on c-Met gene amplification against lung cancer, lymphoma, and esophagus cancers. So this is where we have to see, even though the first compound, the first one, activity and potency, still it had some problems with pharmacokinetic or drug likeness features. Then they were able to identify which region had these issues and they were able to optimize it and come up with a final drug molecule. Let's look at the second case, the B structure, which is axitinib. And this was approved by FDA in 2012, which I already mentioned, for advanced renal cell carcinoma, right? So here what they have done is that during the development process, the crystal structure of phosphorylated construct, which I showed you a PDB, that has been resolved the structure with the inhibitor with a specific activity complex and which had a very good robust structure activity relationship, which provided the importance of rational drug design. But here we also have to see it generating pyrosols 3 benzamide 4 as a starting point for the drug design, where the further effects we were able to make derivatives, because leading to the generation of indazole compounds as novel kinase inhibitors, we were able to come up with compound 5. As you can see, the compound 5, which is in the center, it has with a sterile functional group at the third position of indazole ring was identified to exhibit potent inhibitory effect better than the earlier one, and with a higher level of lipophilic efficiency and ligand efficiency. So this was where we came up with optimization also further to which is bound to the protein structure, and it demonstrated benzamide 4 and the indazole 6th one. So benzamide is the 4 and in the 6th structure indazole, that an additional amide group on the orthosteric site of S-phenyl group would help to make the two important hydrogen bonds. And these two important hydrogen bonds, which I was referring to, the two amino acids, that was mandatory. So in order to make that the interactions possible, they optimized the structure and they finally got into a strategy to get generated axitinib. And this was a remarkable improvement with the cellular potency, desirable physiochemical and pharmacokinetic properties. So this is what I was telling you in my first slide itself. We all think that we can decide which compound to go for animal testing and others. No. So that this slide actually proves that. So basically here you can see that that particular optimization improved the key interactions with those two amino acids. Then only this compound was able to work as expected with better efficacy, lipophilic as well as ligand efficiency with optimal pharmacokinetic properties. And finally, when we are going with heat shock proteins, which I was talking to about the final one, the drug. So here, the biological functions are already being identified. The crystal structures are available. But when you're looking at the compound, we had, through the high throughput screening, we identified that first compound, right? The compound number 7 under the C scheme. So this was completely identified based upon high throughput screening. Okay. But we wanted to look at certain functional groups which could improve the pharmacokinetic property. Moreover, replacing the methyl group to an amide group, changing pyroxyl ring to isoxyl aromatic ring, and modifying some other chemical groups led to a potent effect in animal cancer models. And that's where it is down to a strong one, which is called as limnesibib, which is a drug on. It has gone to the last phases right now, and which has exhibited a potent anti-tumor activity in lung adenocarcinomas targeting EGFR exon 20 insertion mutations and better results in cellular models in the confirmatory clinical trials. So this is the beauty that you have to see how you can implement computational tools to understand where to make changes. And if I ask a question to chemists, medicinal chemists and pharmaceutical chemists, do you know that you have to make these changes in the functional group or where to make the changes in the function to get the interactions? You need to have some visualization and you need to have some understanding about computational interaction studies and many more. So it's a goes in hand parallelly. When you wanted to do some better things in experimental computational studies can enrich its understanding or making decisions. That's why I was telling you guiding decisions. So here we were able to make a decision. This is the region or this is a particular functional group I have to optimize in order to enrich its activity as well as improve its pharmacokinetic property. So successful application of computational methods in anti-cancer drug, these are the best three examples. This is published, I have taken from a literature, and also they have conducted lead optimization, high throughput screening, pharmacokinetic optimization, and activity enrichments were being carried out. These are the few of the FDA approved drugs based upon computational studies and where they have implemented lead optimization techniques also. There are a few number of them. I just listed only a few of them here. Now, this is something very interesting. I'm sure many of the attendees will be working on natural products or extracts from plants. This is a trend and this is very much need being we are in India, right? So these are some of the plant extracts that already being reported. Now the question is, generally when we synthesize some compounds or when we extract some compound or isolate some compounds from crude, always when I was doing my research, the top 10 list for bioassay studies or in vitro studies was anti-inflammatory, antifungal, antimicrobial, then anti-diabetic, anti-malarial, but then later it came to anti-cancer. But the question here is, how do we know that? Whether this compound will really fit in there. If you have money, of course, you can do high throughput screening. You'll try to do all these targets and then see which one it fits in. That's what we call it as high throughput screening because we don't know which one works well. That's the reason why we have to look at implementing computational approaches, which is something called as systems biology or network pharmacology methodology. I'm sure you would have heard about it. I'm not talking too much into this slide, but rather I'll move on to the next slide because we wanted to know how to identify this target. For example, you synthesized a new molecule or you extracted a compound or isolate. You don't know on which disease you have to target. Maybe you like working on malaria, you will test on it, but it might not work. You wasted your time on your wet lab experiments. So it's better that to have an understanding, is there any existing data already available to similar compounds or on the same extract or on the similar functional groups, scaffolds. So for that, we can always use network pharmacology methodologies. And we try to predict the hub genes and we try to look at the active site prediction and try to identify them. So what we are trying to do is we use the smiles of a compound, whether it is an extracted compound or whether it is a synthesized compound, and we try to see what are the possible target genes already being analyzed in this domain. If that structure is not being analyzed yet, we could at least look into similar structures. And then we try to look at what are the partners, what are the interacting partners of the target gene. And we also try to find out what are the different diseases that is associated to that gene. When we combine all them together, that's where we try to identify the possible targets. And let me tell you, this might not be true because the data source should be also relevant. If the data source are all experimental for whatever data that we received, it's somewhat reliable. If the data source is completely based upon a text mining or search, we have to take a much care. And that's where we have to finally go for target prediction. When you have a 3D structure of the protein, you can always go for a structure-based drug design where docking is involved. But when you don't have a structure, now you might say, I can do homology modeling. No, it's not accurate. It's not good for drug discovery projects. But if you have a very good identity or the quality of the model is good, yes, you can. Otherwise, you cannot use it. So we have to see what are the different alternates and how do you select those structure, which region, which amino acid is important for this functionality or for the specific cancer, all has to be looked at. To give you some heads up, these are the databases or websites that you can look at for uh, identifying the possible targets. For example, Swiss target prediction, and then you have string and stitch databases available. You have SEA tool available, Farm Mapper and Chem Mapper, and these are all different tools which helps for the target identification. But as I said, network pharmacology, which you have to do, which we have to do ourselves, where we try to take the compound styles and we try to identify the possible target genes of the similar structure or of the same structure if it's already being explored. And then we try to get the disease associations with this gene and we try to form a network. As you see in the slide, this is a network that is being formed. You can see many of the nodes have been connected to a single one. That single one is a compound, but it is being related to many other genes, and that genes are related to many other diseases. So this is what we call it as network pharmacology, and this can be achieved by using a freely available software called as Cytoscape. There are a lot of tutorials available on YouTube and others, so that I'm sure it will be helpful for you to start. So data collection is something very important, and the source of the data is also very important to make decisions here. So this helps you to identify the target identification. But then we need to have some molecules also, right, to explore, to discover, or to identify where to start. As you all aware, there are many drugs in the market which was derived from plant extracts. I don't want to say examples, there are many of them. But in order to start with natural products, you have a database, something called as coconut database. I'm sure Abhijit was extensively using this during drug discovery hackathon and many of his other projects because we have been interacting with this, especially for these databases. So here we have more than 4 lakh molecules which are natural products and it can be extracted. The problem with the natural products is that many of them are bulky. They are flavonoids, huge, lot of aromatic rings, very lengthy chains. Sometimes this will not fit into the pocket. So make sure you try to give the right molecular weight, right size, right number of sugars, right number of aromatic ring to filter them and try to do your molecular docking analysis. Then everyone's famous Duke's database, which is now being ported to USDA. Earlier it was by itself, it was a database, but now it is ported to USDA. Then from India, we have something called as Impact database, which is from Institute of Mathematical Science, Chennai. And the good thing I like in this database is that they also already try to predict for this particular extract or isolate what could be the possible gene target genes. Because I was telling you, stitch string database can be used. Same is already incorporated into this database. And you can see on the right hand side, they will try to tell you what are the possible target for this particular compound or natural extract. And there's a dedicated database which is called Naturally Occurring Plant-Based Anti-Cancerous Compound Activity Target Database, that is called NPACT. That is called NPACT. And that is what it actually relates to the different cancer types, and also it also different class of compounds also as being considered here. So it will be easy for you when you are starting working on any anti-cancer or cancer projects. So before I continue with a little more methodological aspects, please let me know how much more time we have. It's already 12:15.
Sir, you can continue, please.
Okay.
Okay, sir.
So if you have any questions, yeah, as Abhijit mentioned, you can type it into the this particular chat, or you can stop me in between. If you did not follow something, yeah, you can ask me. I'll try to explain it in a different way so that at least whatever is covered is taken care. So moving forward, we will look at the drug discovery and designing. For example, that particular single slide, how did we do it? What are the techniques did we use to make that optimization or the decision in which functional group to be modified? Yeah, that's what we are going to look here. So normally we understand docking, molecular docking means lock and key hypothesis. That means single key for a single lock. And this hypothesis was pinned down in 1894 by Emil Fischer. But currently, right now, at this moment, 2021, it's only half true. So we have to see that now we are talking about drug repurposing and drug rescuing and drug repositioning. So for a single key, we can have multiple locks. That's what we are trying to explore. So I want to just ask a question right now to everyone. How many of you know what do you mean by drug rescuing? Are you aware of this terminology? I'm sure everybody is aware of drug repurposing, but are you aware of the word called drug rescuing? I'll wait for 1 minute. No? Okay. How about others? Hearing for the first time? Our ICMR is working on that. They have some of the institutions have already published papers on drug rescuing. Okay, I don't hear anyone. I think everyone is hungry and no one is ready to interact. So drug rescuing is mainly some of the drugs fails in the like in the phase 2 trials, right? So there are chances and they don't continue with that. There are chances other research groups take over this compound and then they try to rescue this compound to use it for diagnostic medicine. So there are therapeutic medicines and diagnostic medicines, right? So when you're trying to rescue a drug and trying to understand whether this can be used for a diagnostic purpose, or even if they have taken out all completely, they try to optimize, a different research group try to optimize, make further modifications and try to see still whether it can be worked or not. So we already know what are the challenges in that particular compound. So we have to come up with that. So this is where different diverse people join together. They can come up with the new ideas and new perspectives. And that's where you're trying to rescue a drug. Drug repurposing is what already marketed drug. You're trying to repurpose it for a different disease or a different target. But rescuing, something failed, we are trying to bring it up. So I know two ICMR institutes already working on drug rescuing and very well, they are almost near to prove the optimization is working well. And that's where we have to see how molecular docking is to be looked at. Molecular docking is not simply just giving a score or giving fancy pictures alone. It is about understanding that target in a deeper manner. Molecular docking, not only understanding the interactions between a protein and a ligand, but it is also understanding where or what or which is the bioactive conformation of that particular compound. Once you have in order to find out the bioactive conformation, we are using molecular docking studies. So we need to find out the bioactive. Now, when I say bioactive conformation, what is that? For other people who does not know about conformations and others, or not heard about conformations and others. Basically, when you have a compound in a room temperature, room pressure, it will be having its own shape or molecular crystal shape, very layman term. Otherwise, we call it as a geometry or conformation. But
trying to bind or when it trying to form a co-crystal with a biological complex, that is macromolecule, it could be protein, it has a different shape. The same drug molecule that we have consumed has a different shape. So docking is a process to identify that particular, at what shape it can go and bind to that protein. And that shape is called as bioactive conformation. So molecular docking is a process to identify the bioactive conformation of a molecule. And that conformation is not that easy to form because we need to know that that conformation can form a co-crystal or not. If it cannot form a co-crystal, whatever the docking score you're saying has no meaning at all. So our evaluation of which could be the best structure can form co-crystal is beyond docking score. And that's the reason why we try to perform molecular dynamics and torsional analysis. So if you wanted to derive some data out from docking, these are the following questions that to be answered. Where the ligand will bind? How will it bind? How strong it can bind? We might think, yeah, if it has a dry hydrogen bond, it's all good, it's strong. No, hydrogen bonds are weaker bonds. So we need to see how stable this compound can comfortably sit within the pocket. Why this is interacting? What makes a ligand bind to the receptor better than the others? And like this, many questions, if you can answer, your discussion for the molecular docking will populate, which is called as mechanistic driven molecular docking studies. Then only you can talk about whether your compound is agonist or antagonist. No billion-dollar software can tell you that whether your compound is antagonist or agonist until unless you understand the mechanism of the target that you have chosen. So on the right-hand side, you can see a picture that I have taken from enzyme methodology book. And this is, I'm talking about the enzyme basically. How you can elucidate or extract data when you're before doing molecular docking on enzymes. First, you have to understand the catalytic function. Which amino acid or which is the catalytic site, functional site, domain, motif region. Then only you will be able to identify which is the key amino acid I have to target for interaction. Second, regulatory mechanism. Am I targeting the active site or the allosteric site? And this is very trivial. And the third one is the cleft detection. That means pocket. The pockets are detected by many tools. All these tools are predicting based upon geometry. Does a biochemist agree with that? I'm a biochemist. Basically, I can agree a pocket only when that pocket contains the key amino acids which can upregulate or downregulate when some foreign particle like a drug molecule comes and interact with my enzyme. Otherwise, that pocket, whatever is identified, is of no meaning. Software algorithms try to predict the largest volume pocket as the first pocket. But we don't know whether that contains a biochemically viable amino acids to interact. The next point is geometrical analysis. We need to understand the active and inactive conformation. At what conformation it's active and what conformation it's inactive. Then we need to understand the flexibility. Are you targeting a substrate bound site or an apo site? These informations are all very trivial to make the discussion of molecular docking studies. And then conservation study, very, very important when you're working on cancer related research. So basically conservation means the 3 S: selectivity, specificity, and sensitivity. That means, let's imagine you're working on, let's say, skin cancer, and you have a molecule and you did a cell assay and you found that that molecule is very good for that inhibition for skin cancer. But then you send it to me for review and I'm asking a question. Very good. How confident you are that this compound might not also go and interact with other cancer cell lines? Have you tested it? The blind answer is no, because you have tested only or you focused only on skin cancer. So conservation study has not been carried out. So we need to understand how selective your compound to that specific cell line or target. And that's very crucial. So NCI US has a program called as TPT program, therapeutics program. What they do is you can send 2 compounds to them free of cost. Of course, you have to take care of shipping charges. And they do the cell line studies on 60 different cell lines. All these 60 different cell lines data will be given to you. That's where you can understand the conservation study. The similar conservation study can be done using docking also. You will try to take the different targets from different cancers, targets, and then you try to do docking and understand the selectivity and specificity. So without doing this conservation studies, whatever the results that we are giving, it's really, really basic. It's just a starting point. So it's very important how we can add a value for the translation of the data that we are generating. And finally, we have to look at the surface characteristics like solvent accessible area, lipophilic regions. Earlier, I discussed that the optimization was mainly focused on lipophilic efficiency, right? And then finally, we talk about energy. Now, you might think, I thought docking was so easy, just getting the score and pictures, but you said many things. Yeah, this is actually what we are talking about docking. So we have to change. If people were already being published in Nature without telling all these things, that is old story. Now we are going to go with a competitive era now. Let's think about the next 5 to 10 years. It's going to be more challenging. You need to come up with a lot of new perspectives. Now, surprisingly, this book was published in 2008. So it's an old story, which we might not have used it yet. So it's very important that how we look at these computational tools and what data that we can extract it from them. That's why I said, if you do computational studies in the right manner, you can get decisions. So I think it's already 1 o'clock. So please let me know. Otherwise, I'm done for the session.
Hello, sir. If you have some more to share, you can share. We will wait. No issue at all.
Okay, okay. I will just want to quickly tell you one more slide and then I will wind up.
Of course, sir. Of course.
Just let me. Yeah, this was the main one. So when we look, so whatever I was trying to tell you was objectives of a drug design. When you come to drug discovery, so whatever docking I was telling you, when I was talking about 3 different anti-cancer drugs, I was telling you about the interaction with the key amino acids. So that was the technique I was discussing so far. Now I want to discuss about the pharmacokinetics, because we had the third one which was having a poor pharmacokinetic features. That's why the initial compound was, even though it was active, we did not consider that. But later they made the improvements, optimization, and they got the pharmacokinetic properties. So let's look at the objectives of the drug discovery or lead discovery. For that, it is we have to know 3 different terminologies of small molecules. One is hit, a lead, and a drug molecule. The hit molecules are mainly few thousands of molecules which shows potency or activity or understanding the binding towards the target. That is what we call it as hit. And we are looking at a very micro level. We are assuming that the compound has already reached the target and it is interacting comfortably. But the next question is, will it reach the target comfortably? Right? If you want to go from Kanyakumari to Delhi, as in the next second, will you reach Kanyakumari from Delhi? No. You have to go through, there's a time, you have to take food, you have to rest. A lot of hurdles are there. The same way, when drug being administered orally or some other administrative routes, you have to see whether the drug can safely reach your target without it affecting much or hurdles. For example, we have to understand whether the drug gets changed or modified by the metabolic pathways or organs, or whether the drug is going to affect your organs different cycles. This is where we look at how the hit molecule transforms to a lead molecule. So in order to transform a lead hit to lead, you need to see well how far the compound is safe. We never expect that it shouldn't have any toxicity, but admissible ones or permissible ones. And then absorption rate. Oh, sorry. Can you see my screen?
No, sir, it's not visible now.
Is it possible? Is it visible now?
Sir,
Okay, let me
Part of it
reshare it
is
again.
visible. Part of it is visible now. No, again it is
Let me
not visible.
reshare it. Let me reshare it. Okay. Is it visible?
No, sir, not yet. Yes, it's visible. It's visible now,
Oh,
yeah.
okay. Oh, okay, fine. So we have to also see toxicity, the intestinal absorption rate, metabolic stability, whether when it passes through liver metabolism, we want to know whether this compound will be stable there or it will form a metabolite. Or we also have to look at solubility, permeability, flexibility, etc., etc. So, including potency, if that particular compound has an optimal balance of all the other pharmacokinetic and pharmacodynamic properties, that's where we call it as a lead molecule or a drug-like molecule, where the drug-like properties will be there. Once all this is done, and we go for a IND filing or FDA approval, that's where we call them as a drug molecule. Until then, we call it as a drug candidate. So we need to identify the molecules which has a balance of all these properties. Unfortunately, most of the time we publish basically looking at potency studies done, maybe a cytotoxicity studies. But we have to extend and we have to go through all this data. Then only the pharma industry will be getting attracted to your patented molecule because they need these data to be looked at. So we need to quickly identify such situation when that particular balance is not possible. It's very trivial. If you're not able to get that balance, it's better to fail fast so that you fail cheap because you don't have to spend money. And that's exactly the reason why there's an increase in the medicine because they were not able to identify the failures much in earlier stages. There are many cases where after getting into the market, it has been retracted, revoked from the market after 1 month or 3 months of usage. It's a huge loss for the company. So later they do optimization. When it comes to the market, the cost increases. Only when confident, move forward with certain molecules and try to avoid missed opportunities. Unnecessary usage of different filters or wrong strategies can lead to missed opportunities. That means some compounds might be very important, but you unknowingly or by mistakenly, that compound is not there in your decoys or in your subset, which will be going for further analysis. So by that, I would like to stop the session and I'm happy to take any questions. And these are some of the publications that came out from my group, considering all these factors. And yeah, the scientific report, that paper we worked on EGFR, of course, using Autodoc. So nobody questioned us why you didn't use paid journals, the paid software, nothing like that. So, and others, I am also working on longevity and aging. So on the left-hand side, mostly it is on the aging related targets and
Excuse me, sir.
anti-allogeneic.
Sir, actually the sir, next slide is not visible, Mr. About the publication slide. We are right now observing the 74th slide. Objectives of lead
Let
discovery
me
with title.
let me
I
reshare
think
it.
right, sir.
Is it visible now?
Sir, I think no, sir.
Oh, okay. What I will do is, I hope everybody have access to the chat, right?
Yes.
Yes, sir. Huh.
I'm I'm pasting a screenshot there,
Right,
so
sir.
everybody gets it. Hope you got it, right? Yeah, if my screen is not visible, these are the only 2 slides left. So I just shared it in the chat. Okay, so I am done. A quick overview
Sir,
about some case
later on,
studies.
if we have your means a brief write up on your lecture, sir, we will share with the participants who are interested.
Absolutely.
Sir, I think your slide is also available in slide share if I'm not
Yes, sir.
wrong.
Yeah.
So
Yes, you
participants.
are right. So
Available
let
in
me
chat
let
box
me.
also. Chat box also, it is now visible.
So today whatever I have presented, the slides are available in this particular link. I'm sharing it in the chat box. So you can take those slides. Yeah. Yeah. Okay. So any questions? I know everyone is hungry, so we can, if no questions, we can take it up later also. Yeah.
Thank you, sir. I request to all the participants if they have any queries related to the drug discovery or the sir's talk regarding various methodologies of computational drug discoveries related to oncological products also, they can ask or type in the chat box. Sir, you delivered very nice lecture, very nice presentation, and we gained a lot from your topic, basically the M Pharmacy and PhD students working on that. You discussed a lot and about the computational methodologies, high throughput screening models. Mm, Just we can wait for a few more minutes so that some questions may be able to see.
Thank you so much, sir, for a very informative speech on computational approaches in anti-cancer drug discovery and precision oncology and lead optimizations, various lead optimization schemes also, and so many new approaches to think about nowadays. Sir, I am thankful to Mr. Abhijit also, who is a big fan of yours and who has actually helped us to contact you. Anyway, sir, we will have you in future also time to time. Hope so. Sir, I think you have also enjoyed your lecture. So thank you so much, sir.
Absolutely.
Yeah.
Thank you. Thank you so much for your patience and for this opportunity. Hopefully, things will slowly work out and people will be able to work on this similar fashion.
Of course.
Thank you so much. Best
Thank
wishes.
Excuse me, sir.
Yep.
Yeah, sir, with your permission, I want to share with our participants regarding the podcast where you actually periodically share about all the drug discovery stories in a very nice format. So, can I share the
I
link
shouldn't
with our
market
students?
myself. I shouldn't market myself, but go ahead.
So, can I share the link of your the
Please,
podcast with the?
please, please, please,
Okay.
please.
And sir, this is
The
really
podcast, yeah, please go ahead, Abhijit. Yeah.
actually, in our India, also in our all over the world, there are only few scientists or few people are doing such kind of podcasts, especially in drug discovery. We know scientist Vincent who is doing podcast in viruses and other various neurological diseases, but you are the one who is doing very nicely on drug discovery. So I've shared the link in the chat box. Participants can click on this link. They can go through the podcast. There are lots of stories, sir, you have shared. So I think it will be very great inspiration for the young people to know about the stories of drug discovery. And we are deeply thankful to you, sir, for doing such great work for us.
Thank you. Thank you so much. You guys are all motivational to me because my aim is just transfer of knowledge. And that I try to find out which mean we can transfer. And these all, whatever being shared, it's all stays there ever and ever. So let's see. But it goes on, keep on changing. Next 10 years, whatever I'm telling now, it's going to change. That's how it is.
Right, sir.
Yeah.
Thank you so much, sir.
Thank you, sir. Have a nice day. I am sure we will be very much enriched by the speeches of our today's eminent speakers of Atal FDP. I also extend my heartfelt thanks to all the delegates, faculty members on behalf of NIT Pharmacy Institute and their management. See you tomorrow at 10 a.m. Have a nice day, everyone.
Thank you, Dr. Soumya. Thank you.
Thank you, ma'am.